Hello everyone, I am Dr. Suman Bhandary, Assistant Professor, Department of Biological Sciences, School of Life Science and Biotechnology of Adamas University. Today, we will be discussing on the module Isolation of Industrially Important Microbial Strains and Fermentation Media. This lecture series constitute of 3 lectures, where at first we will discuss about the content of today's lecture. The content of today's lecture is sources of industrially important microorganisms. And next to that, we will try to identify the methods for the isolation of industrially important microorganism. Let us discuss about the learning objectives. So after the end of this lecture, you will learn about the diverse sources of industrially important microorganisms. And also you can recognize the role of these microbial strains in different industrial processes as well as biotechnological applications. And at last, we will discuss about the enrichment culture techniques along with the other screening as well as isolation methods and how these methods are applied to isolate specific microbial strains. Let us start with the sources of industrially important microorganisms. So generally, we know that industrially important microorganisms can be sourced from various natural environments as well as human-made processes. So some of the common resources or common sources has been mentioned here in this through this flow diagram. There you can see that few are natural sources, some are man-made sources or human-made sources, and some we are getting from the non-conventional sources, mainly mainly those microorganisms are known as extremophiles. So if you look into the normal sources, here normal sources means we can find microorganisms from soil, we can find microorganisms from water as well as from air. You know, in soil, we can find actinomyces, mostly streptomyces species. And these species are well known for producing different different antibiotics. There are various fungi that is present in the soil that contribute to the production of enzymes as well as several bioactive compounds. If you check water, in the aquatic habitat, we can have several aquatic bacteria like Pseudomonas and Bacillus species. And these are often found in water as well as that are used mainly in the bioremediation process. Along with aquatic bacteria, there are some microalgae that have application in biofuel production as well as production of bioactive compounds that are used in pharmaceutical industries. Now from soil and then water, then if you enter into air, here you can see that certain bacteria, which we call as airborne bacteria, like Bacillus species, that are commonly found in the air and have industrial applications. Some fungal spores. There are some fungi which release spores into the air, and these have industrial uses. Now coming to the human-made sources. If you look into the fermentation processes, there you can see that decades back, if you look back to 17th or 18th century, that people have started using Saccharomyces cerevisiae, and still we are using it widely in the fermentation industry for the production, for brewing as well as for baking. We are using lactic acid bacteria, mainly lactobacillus species, in the dairy industry for making dairy product as well as for making pickles. There are certain microorganisms that are used in the bioremediation processes, which we consider as bioremediation microorganisms. Bacteria like Pseudomonas, Bacillus species that are used for the cleanup of the industrial waste as well as the pollutants. Along with that, we can use some anaerobic bacteria, mostly the methanogenic bacteria. And these methanogenic bacteria are used for the treatment of organic waste anaerobically. And they are digesting this organic waste anaerobically. The third category, that is microbes from extremophiles, we can consider thermophiles, psychrophiles, halophiles in this category. Thermophilic bacteria like Thermus aquaticus, we all know that Thermus aquaticus is a rich source of enzyme, mainly known as Taq polymers, which is used in polymerase chain reaction. Along with that, there are some halophiles, some halophilic archaebacteria that are studied for their enzymes, which have industrial applications. In bioreactors, mainly used in fermentation as well as in pharmaceutical industries, microorganisms adapted within the bioreactor to specific industrial conditions, sometimes naturally, sometimes we are modifying these microorganisms genetically using recombinant DNA technology. And this technology has been used for the development of genetically modified organisms, microorganisms, mainly for specific industrial processes. Not only that, in medical as well as clinical samples, some pathogenic bacteria, which are now used in the production of vaccines, some yeast and molds are used for the production of antibiotic as well as pharmaceuticals. So now the question will come to our mind that from these three sources, we have seen that there are several bacteria, several microorganisms, not only bacteria, fungi, algae, that can be isolated, that can be screened, which has relevance in different industry. Now, we need to know what type of microorganism we can consider as industrially important microorganism. Means what kind of desirable traits should be present inside those microorganisms, then we can consider those microorganisms as industrially important microorganisms. So a microorganism used in an industrial process must have other features besides just being able to produce the substance of interest in high yield. The first and foremost, the organism must be capable of growth and product formation in large scale culture. Means if you are scaling it up from lab scale to large scale, the microorganisms should be capable of grow as well as produce the desired product in large scale. In case of fungi, in case of yeast that has industrial applications, it should produce spores or some other reproductive cells from so that uh, it can be easily inoculated into the large culture vessel used to grow the producing organism on an industrial scale. Now, the third characteristics that it must also grow rapidly and produce the desired product in a relatively short period of time. And it must also be able to grow in the liquid culture medium obtainable in the bulk quantities at a low price. Means it can means we can grow the microorganisms in a large scale in a cost-effective medium. And we also consider an industrial microorganism should not be pathogenic, but not all the cases, because the production of vaccines, there we are using pathogenic bacteria. But mostly we consider industrial microorganisms should not be pathogenic, especially to the human or economically important animals and plants. And also, they will never produce toxic metabolites, which may harm us as well as economically important animals and plants. Because of the high cell densities in industrial microbial processes and the uh, virtual impossibility of avoiding contamination of the environment outside the growth vessels, means outside of the bioreactor or the fermenter, a pathogen would present potentially disastrous problem. And finally, an industrial microorganisms should be amenable to genetic manipulations because increased yields are often obtained by means of mutations as well as classical genetic selection techniques. So a genetically stable and easily genetically engineered microorganism is thus a clear advantage for an industrial applications or for the industrial process. Now, when you are developing or searching a microorganism that have some industrial applications, you may encounter some problem. And these problems often associated with industrial microbial processes. The first problem is uh, the first problem is finding the least expensive medium in which to grow the microorganisms so as to maximize yield and profits. And most of the cases, this is the waste product. And this waste product is coming from another industrial process. And then only the culture medium could be cost effective. Generally, we consider corn strip liquor that is coming from sugar processing industry. We consider whey that is coming from milk industry to prepare the large scale culture medium in a cost effective manner. Next point is maintaining strain purity and developing better strains for improving the yield. Now, a single mutation may decrease the yield by a significant percentage or result in undesirable substances being produced. So the industrial research laboratories constantly seeking better strain for the production of their product and for better yield. The third problem is preventing contamination by other microorganisms and mostly by viruses, mainly phage viruses, because that live on the microorganisms that we are involving in fermenter for the industrial processes. That is why the culture media we are using here must be sterilized prior to being inoculated with the desired organism, and purity must be maintained throughout the process, throughout the production process. Because a small quantity of contaminant may produce an enzyme that can destroy the product in thousands of gallons of mediums. And for many microorganisms, viruses present a constant danger, as because a single phage virus can infect and can destroy the desired microbes of the entire tank. So sterilization of large containers and huge quantities of media represent both an engineering as well as microbiological challenge. Last but not the least, developing rapid and efficient methods for purification of the desired product. Why? Because we have to ensure safe use of the product and stability of the product. So the product or the products of many fermentations are often unstable if they are present in impure form. Or means in most of the cases we have object there, if they are present in impure form, they are subjected to unwanted modifications. If they are not purified quickly. So as every step in the purification results in a loss of product, we need to ensure that there should be such type of microorganisms that we have to develop or we have to search from the environment, from the natural as well as man-made sources, which can produce lesser number of byproduct. Then our downstream process would be easy, would be cheaper, would be less time consuming, and less manpower will be involved there. And the last point is that always striving to improve yield by modifying the strain. So we have to involve constantly in the strain improvement process. As well as we have to improve the quality of the culture medium. We have to check nutrients that is present inside the culture medium, and we have to produce a favorable growth conditions. We have to optimize culture conditions for betterment. Like we have to optimize pH, we have to optimize aeration rate, we have to optimize dissolved oxygen concentration, etc., etc. Now, there are certain microorganisms that are used in industry. And here's a list. In the first column, you can see the type of microorganisms. where we have listed photosynthetic bacteria, yeast, lactic acid bacteria, actinomyces, fungi, etc., and their applications, and what kind of modifications we can perform, what kind of specification we can include for their better growth yield. Now, after that, we are gradually moving to the isolation process. So far, whatever we have discussed here, we came to know about the sources. We came to know about the desirable traits of the microorganisms that should be considered as industrially important microorganisms. We have discussed about the problems that we can face during this development process. And now we will discuss about the isolation and then the screening techniques. So, isolation of suitable microorganism from the environment, we have to adopt few strategies. Here we will address two strategies. One is shotgun approach and another one is the objective approach. So first we will discuss about the shotgun approach. In the shotgun approach involves collecting samples from diverse range of environments without specifically targeting a particular microorganism or a group of microorganisms. So generally in this method, typically we are including or this method typically includes obtaining samples from different locations. It could be soil, it could be water, it could be air, it could be industrial settings. Without any prior knowledge, without any background knowledge of the specific microorganism of our interest. Now, this technique has some shortcomings and some advantages of it, definitely. So the advantage of this shotgun approach is exploratory and may uncover novel microorganisms with potential industrial applications. As because it allows for a broad survey of microbial diversity. Now, the disadvantage of this process is it may result in a large number of isolates and many of which may not have industrial relevance, may not have any industrial applications. And the second disadvantage is that sorting through a diverse group of microorganisms is time consuming. So from shotgun, we are moving to the alternative strategy, that is objective approach. So in the objective approach, it involves a targeted strategy, focusing on specific criteria or characteristics that the desired industrial microorganism should possess. This method starts with the background information, with a clear understanding of the industrial requirements and the traits needed in the microorganism. Means specific metabolite capabilities, tolerance level to certain conditions we have to address here. And then we will go for sampling. Sampling is then conducted in environments where such microorganisms are likely to be found. Means here we are not going in, we are not searching in various fields. So this typical approach has certain objective, certain advantages. That is, it is more focused, leading to the isolation of microorganisms with traits tailored to industrial needs. And it may save time and resources compared to a broader shotgun approach. But the disadvantage of this process is there is a risk of overlooking potentially useful microorganisms that do not fit the predefined criteria. And also it requires prior knowledge about the characteristics of the desired microorganism. So the second approach is enrichment culture. So once the sample have been collected, a major problem is deciding on the growth media and cultivation conditions that should be used to isolate the target microorganisms. An initial step is often to kill or repress the proliferation or repress the growth of common microorganism which is present in the same population, which is present in the same microbiota, and also encouraging the growth of the rare microorganism that we are targeting as a microorganism with industrial importance. So enrichment culture may then be performed in batch culture or often more suitably uh, in continuous system. So there we can adopt some techniques. In table 1 here, you can see that the left-hand side, we have mentioned the type of organisms. And the right-hand side, we have mentioned the culture conditions. So for thermophiles, we have to maintain high temperature starting from 42 to 100 degrees centigrade. For psychrophiles, we have to maintain low temperature. For acidophiles, we have to maintain low pH. For halophiles, we have to maintain high salt concentration, so and so on. Now, after this enrichment isolation, the next step to enrichment and isolation is screening. Now, in the screening process, first we have to understand about the definition of screening. What is screening? So screening can be defined as the use of highly selective procedures to allow the detection and isolation of high yielding species of microorganism of our interest from among a large microbial population. Thus, to be effective, screening particularly allows the discarding of many valueless microorganisms, while at the same time allowing the easy detection of the small percentage of useful microorganisms that are present in the population. So the concept of screening will be illustrated by citing specific examples of screening procedures. that are basically utilized in industrial research programs. Generally, we apply crowded plate technique. Later on, we will discuss this technique, elaborately. So here we can see that screening is subsequent isolation of certain microorganism means we are developing a pure culture on a solid culture medium, a solid growth medium, mainly by using agar medium. And there we are using selective culture medium for the development of pure culture. And this pure culture then must be screened for the desired property, like production of specific enzyme, like production of specific compound, like antibiotic, etc. So, if a microorganism that has been isolated and screened from a particular source, and we are getting a pure culture, but the microorganism should have some characteristics, then we consider it as an ideal industrially important microorganism. So what kind of characteristics it should have? The first one is genetic stability, then efficient production of the target product, less by production of byproducts, limited or no need of supply of vitamins and other growth factors through culture media, utilization of a wide range of low-cost culture medium available as carbon source, amenable to genetic manipulation. They should be safe to use, non-pathogenic, will not produce any toxic byproduct or toxic product, and ready harvesting from the fermentation. Means this should be larger in size so that we can harvest this microorganism quite easily from the fermenter vessel. Now, the screening of microorganism for new products. So, using the techniques, a set of highly selective procedures that allows isolation as well as detection of microorganism producing the desired metabolite. The process is known as primary screening. Now, what type of method we use here in this primary screening approach? So we will use those methods which are rapid, which are inexpensive, which are specific but effective, and which are predictive. And here, this primary screening is the most critical step because in this step, we are eliminating a large bulk of microorganisms. We think that these are unwanted microorganisms or useless isolates, which are either non-producer or which are producer of known compounds. Now, we will discuss about the types of screening. So, here we will discuss about the two types of screening techniques: primary screening and secondary screening of microorganisms. The economics of a fermentation process largely depends upon the type of microorganism used. If the fermentation process is to yield a product at a cheaper rate, the chosen microorganism should give the desired product in a predictable as well as economically adequate quantity. Now, the microorganisms with the desired characters is generally isolated from natural resources like soil, water, or any other man-made resources that have certain biochemical characteristics, and it should possess stable biochemical characteristics. And it should grow well in ambient temperature. Along with the other traits that we have discussed earlier, there are certain other characteristics must be there to consider it as a producer strain. So it should grow in ambient temperatures 30 to 40 degrees centigrade. Because if you grow that microorganism at that particular temperature range, means you can reduce the cooling cost. You can also reduce the heating cost. And it should possess minimum reaction time with the equipment used in the fermentation process. And it should yield only the desirable substances without producing undesirable or byproducts, undesirable substances or byproducts. And it should possess optimum growth rate so that it can be easily cultivated on a large scale. Now, there are multiple techniques we can apply here. So first, to discuss with the primary techniques or primary screening techniques. So primary screening is basically a process for the detection and isolation of microorganisms of our interest. But this technique does not provide much idea about the production, does not provide much about the yield potential of the microorganisms. It separates out only a particular microorganism or a few microorganisms from a diverse group of microorganisms, which we consider as valueless. Nothing else. So the elementary steps that are performed to select the desired microorganisms and eliminate the undesirable microorganisms are basically termed as primary screening. There are several techniques we use in this primary screening approach, which includes crowded plate technique, oxanography, enrichment culture technique, etc., etc. Now, here we can say that the crowded plate technique, indicator dye technique, enrichment culture technique, oxanographic technique, and techniques of supplementary volatile and organic substrates. Now, one by one, we will discuss about each of the techniques. So first, we will discuss about the crowded plate technique. So what is crowded plate technique? This technique is primarily employed for detecting those microorganisms which are capable of producing antibiotics. And this technique starts with the selection of a natural substratum like soil or other source containing microorganisms. And then we perform serial dilution of the source material. After that, a suitable alicot of the serial dilution is chosen, which is able to produce 300 to 400 individual colonies when plated on an agar plate. And after incubation, you can see this number of seed colonies over the culture plate or over the surface of the agar plate. So this particular technique is known as crowded plate technique. The antibiotic producing activity of a colony can be indicated by no growth of any other bacterial colony in its vicinity. And the region of no growth is indicated by the formation of a clear and colorless area around the antibiotic producing microbial colony on the agar plate. And this particular clear transparent area or zone is known as growth inhibitory zone. Such a colony that is isolated from the plate and purified either by making repeated subculturing or by streaking on a plate containing a suitable culture medium. Now, this purified culture is then tested for its antibiotic spectrum with some test microorganism. However, this crowded plate technique has limited applications, as it will not give indication of antibiotic producing organism against the desired organism. So this technique has been improved later on by employing test microorganism to know the specific inhibitory activity of the antibiotic. And in this modified process, suitable serially diluted soil suspension is sprayed on the sterilized agar plate to allow the growth of isolated and individual microbial colonies, approximately 30 to 300 CFUs per plate after incubation. And then the plates are flooded with a suspension of test microorganism. And then the plates are again incubated or further incubated to allow the growth of the test microorganism. The formation of inhibitory zone of growth around certain colonies indicates that these suspected colonies have antibiotic activity against the test microorganism. And a rough estimation is of the relative amounts of antibiotic produced by the microbial colony can be estimated by measuring the diameter of the zone of inhibition of the test microorganism. So antibiotic producing colonies are later on isolated from the plate and are purified before putting to further testing to confirm the antibiotic potential of the microorganism or antibiotic activity of the microorganism. The next one is the enrichment culture technique. This technique is generally employed to uh, isolate those microorganisms that are very less in number present in the soil and also possess specific nutrient requirement, but are important industrially. They can be isolated if the nutrients required by them is incorporated into the medium or by adjusting the incubation conditions. Actually, this method or this technique was designed by Beijing to isolate the desired microorganism from a heterogeneous population. Basically, this particular technique consists of the following steps. So first we need the nutrient broth, and the nutrient broth is inoculated with microbial source material and then incubated at a particular temperature. A small portion of the inoculum is then plated on a solid culture medium and well isolated colonies are obtained. Now, suspected colonies from the agar plate are then subcultured on fresh culture medium and subjected to further screening. So this particular technique is known as enrichment culture technique. Now we are going to discuss about the oxanographic technique. This particular technique is employed for the detection and isolation of microorganisms capable of producing certain extracellular substances, such as growth stimulating factors like amino acids, like vitamins, etc., etc. A test microorganism with a definite growth requirement for a particular metabolite is used in this method. And for this purpose, we have to spread a suitable alicot on the surface of a sterilized agar plate and then allow it to grow. Then we can see some isolated colonies after incubation at a particular temperature. Now, a suspension of test microorganism with growth requirement for the particular metabolite is flooded on the above plate containing isolated colonies which are subjected to further incubation. And the production of the particular metabolite required by the test organism is indicated by its increased growth adjacent to colonies that have produced the required metabolite. And such colonies are isolated, purified, and stock cultures are prepared, which are then used for further screening. Now here, for this process, we need to prepare two culture plates. In the first step, we have to prepare the first plate. And how to prepare? Let us discuss the technique through this model diagram. Here you can see. So this is your first plate and this is your second plate. In oxanographic method, first you have to take a petri dish, a pre-sterilized petri dish. Aseptically, you have to remove the lid and then you have to place a pre-sterilized filter strip over the culture plate. Then you have to pour culture medium, and when it is solidified, you have to pour the required amount of alicot from the soil sample. Then you can spread the soil sample, the alicot, over the culture plate, and you can put the lid on and incubate at a particular temperature. So after incubation, you can check several isolated colonies in the first culture plate. Now, in the second culture plate, or in the second step, you have to prepare another culture plate. You have a test culture, and it's a known culture. You know it is lysine auxotroph. Means we have to provide lysine along with the culture medium to grow a particular bacteria. Now you have your test culture. You need a minimal medium that is lacking lysine. You will pour it in a fresh pre-sterilized petri dish, let it solidify, and then spread the test culture and incubate it at a particular temperature. Now, after incubation, you can see that there is no growth of the test culture. Why? As because the culture media is it lacks lysine, and the test culture is lysine auxotroph. Now what you are doing, so over the second plate, you have to place the first plate by holding this filter paper, two ends of the filter paper. And then you are incubating it further. Later on, what you can see that while in the second plate, there was no growth, no colony has been detected. But when you have placed the first plate agar medium over the agar surface of the second plate, now you can see some colonies present here. So here, these colonies are basically growing now because here the soil culture, which consists of several colonies, there are certain colonies which is adjacent to the growth medium of the colony which are now growing can produce lysine, can release lysine, and it is diffusing through the agar medium and it is entering into the second plate, and now the test culture will grow. So such colonies are isolated, purified, and stock cultures are prepared, which we can use for the production of lysine industrially. But further screening process is required, whether they can grow in a large scale or not, just to check. Now, we will discuss about the other techniques. Basically, this technique is known as indicator dye technique. Or sometimes we can say it as a primary screening of organic acid and amine producing microorganism. So here we will incorporate certain pH indicator dye like neutral red, bromothymol blue into the nutrient agar medium. The change in the color of a particular dye in the vicinity of a colony will indicate the ability of that colony to produce an organic acid or a base. So production of an organic acid can also be detected by an alternative method. And in this method, we can use calcium carbonate. Calcium carbonate is incorporated into the agar medium, and the production of organic acid is indicated by the formation of a clear zone around those colonies which release organic acid into the medium. So we can use both the dye, indicator dye, or else we can use calcium carbonate to identify whether the microorganism that we have isolated from a particular source can produce organic acid or not. And the identified colonies are then isolated and purified either by repeated subculturing or by streaking method. And then you can prepare a stock culture for further qualitative as well as quantitative screening tests. So the next one is the primary screening for antibiotic producing microorganism. Before that, we can discuss another technique that is basically used as a primary screening technique of supplementary volatile organic substances. So this technique is employed for the detection and isolation of microorganisms capable of utilizing carbon source from volatile substrates like hydrocarbons. Or we can consider low molecular weight alcohols or similar carbon sources. So what we are doing here, we are doing suitable dilutions of the microbial source like soil suspension are sprayed onto the surface of sterile agar medium containing all the nutrients except the one mentioned above. Means it could be hydrocarbon or low molecular weight alcohol. Now the required volatile substrate is applied onto the lid of the Petri plate, which are incubated by placing them in an inverted position. Now, enough vapor from the volatile substrate sprayed to the surface of the agar within the closed atmosphere to provide the required specific nutrient to the microorganism, which grows and form colonies by absorbing the supplemented nutrient. Now, the colonies will be isolated, purified, and stock cultures are made for further screening process. So here we are simply identifying whether the culture can utilize these volatile organic substances or not. Now, we will discuss about the primary screening of antibiotic producing organisms. So initially we have discussed that the crowded plate technique has been used and the diluted source is spread on the agar plate, and then the organism are allowed to grow in an incubator at a particular temperature. And after incubation, the ability of a particular colony to produce antibiotic is indicated by the development of the inhibitory zone or zone of inhibition around that particular colony. But there is a problem. So initially we have discussed the primary screening is not a confirmatory screening approach. So primary screening carried out by this technique does not necessarily select antibiotic producing organisms. The question is that inhibition around the colony may be due to change in pH of the surrounding medium by the colony and rapid utilization of the nutrient in the surrounding area. So that also can create this transparent area. Because of the deprivation of nutrient deprivation, no bacterial colonies grow there. So further testing again is required to prove that the inhibitory activity associated with a microorganism can really be attributed to the presence of an antibiotic. And that is why we can say that crowded plate technique has limited application. Why? Since usually we are interested in finding a microorganism that can produce antibiotic, that can have antibiotic activity against specific microorganism and not against the unknown microorganisms that were by chance on the plate in the vicinity of an antibiotic producing bacteria. So we have to test those isolated colonies against a test microorganism, which could be considered as an indicator organism to prove the specific antibiotic activity of the isolated microbial colony. Now, we will discuss about the secondary screening approach. So far, whatever we have studied, we came to know that primary screening is usually followed by secondary screening to test the capabilities of organisms which are obtained in primary screening. And secondary screening allows the further sorting of those microorganisms that have real value of industrial importance and discarding valueless microorganisms. So the secondary screening allows determination of the most efficient and suitable microorganism to be used for commercial production of the fermentation product from different potential strain as detected and isolated by primary screening. And mostly secondary screening is conducted on agar plates or in flask or in small scale fermenters containing liquid culture media. Agar plates are not as sensitive as liquid culture, but have advantages in secondary screening because more information we can obtain from solid culture medium. But as because the agar plate, they have limited surface area, they have limited space. So to understand the growth as well as production yield of the test microorganism on a large scale, we need to perform small scale fermenter. And agar culture only provide a limited indication of actual product yield among various isolates. So to obtain information, we must employ liquid culture because it provides us a much better picture of nutritional, physical, and production responses of an organism to actual fermentation production condition. So there are two types of secondary screening technique. One is known as uh, qualitative secondary screening. Another one is the quantitative secondary screening. So in the secondary screening, qualitative secondary screening, it tells us the spectrum of microorganisms which are sensitive to newly discovered antibiotic, but quantitative secondary screening tells us yield of antibiotic, which can be expected when microorganisms is grown in various different media. So certain information we can get from the secondary screening process, the ability or potentiality of the microorganism to produce certain metabolite that should have industrial importance, and we consider it as industrially important microorganism. The quality of the yield product, the type of fermentation required for large scale production, and elimination of the useless microorganism which are not industrial important. So to evaluate the true potential of the isolated microorganisms, both qualitatively and quantitatively, that are generally conducted in secondary screening. And the sensitivity of the test microorganism towards the newly discovered antibiotic producer is generally analyzed during the qualitative analysis, secondary screening process. While the quantum yield of the newly discovered antibiotic is estimated by the quantitative secondary screening technique. Now, to evaluate the potentiality of the microorganism, to determine the product produced by the microorganism, whether it's a new compound or not, to determine whether this microorganism has better yield potentiality or not. What should be the optimum growth condition? I mean, what should be the pH? What should be the aeration rate? What should be the temperature? And it should detect whether the isolated organism is genetically stable or not. Whether it is amenable to genetic manipulation or not. Whether it is chemically stable, means the product it is producing, whether it is chemically stable during the downstream process. Physical stability, chemical stability, biological stability of the product to be screened in this particular secondary screening technique. And through this technique, we should reveal the physical properties of the product, determine whether the product produced by the microorganism in a fermentive process, it is toxic or not. And this particular technique should reveal that whether the product produced in the fermentation process exists in more than one chemical form. If it exists in more than one chemical form, the amount of formation of each chemical formation of these additional products is particularly important, since their recovery and sale as byproducts can greatly improve the economic status of the fermentation industry. But sometimes, if the byproducts are more, then the downstream process, the purification process will be lengthy, will be strenuous, will not be cost effective. So we have to assess at this particular screening technique that if it produces more than one byproduct, then we have to check the economic status of the byproduct. And the newly isolated microorganism should be identified to the species level. And this will help in making a comparison of growth pattern, yield potentiality, and other requirements of test microorganism with those already described in the scientific literature, or else in some patent literature. And lastly, we can say that it should select industrially important microorganism and discard others which are not useful. And the newly isolated strain should be economic or economically used in the fermentation process. Now, we will discuss about the technique that is used in secondary screening. So there are several techniques and procedures that can be used for secondary screening. However, only a specific example of estimation of antibiotic substance produced by species of Streptomyces we will discuss here. And the method is known as giant colony technique. This particular technique is used for isolation and detection of those antibiotics which diffuse through solid medium. So species of Streptomyces is capable of producing antibiotics during primary screening process, and the isolated Streptomyces culture is then inoculated into the central area of a sterilized petri dish containing nutrient agar medium, and the plates are then incubated until sufficient microbial growth takes place. Then cultures of test microorganism whose antibiotic sensitivity is to be measured are strict from the age of plates up to, but not touching the growth of Streptomyces, and are further incubated to allow the growth of the test organisms. And then we have to measure the distance over which the growth of different test organism is inhibited by the antibiotic secreted by the isolated Streptomyces species. With definitely it should be in millimeter. Now, the relative inhibition of growth of different test microorganisms by the antibiotic producer is known as inhibition spectrum. So here we will judge the inhibition spectrum of the isolated Streptomyces. And those organisms whose growth is inhibited to a considerable distance are considered more sensitive to the antibiotic than those organisms which can grow close to the antibiotic. Such species of Streptomyces which have potentiality of inhibiting microorganism is preserved for further testing. Two more methods we can use in the secondary screening approach. The first one is the filtration method and the second one is the liquid medium method. So in the filtration method, it is employed for testing those antibiotics which are poorly soluble in water or do not diffuse through the solid medium. The Streptomyces is grown in a broth and its mycelium is separated by filtration to get culture filtrate. And then different dilutions of antibiotic filtrates are prepared and added to the molten agar plating medium and allowed to solidify. Later on, the cultures of various test microorganisms are strict on parallel lines on the surface of the solidified medium, and such plates are then incubated at a particular temperature. The inhibitory effect of antibiotic against the test microorganisms is measured by their degree of growth in different antibiotic dilutions. So this particular method is known as filtration method. The next method is liquid medium method. Here, this method is generally used for further screening to determine the exact amount of antibiotic produced by the microorganisms like Streptomyces, as we discussed earlier in the giant colony technique. So we have to take a conical flask or Ardmar flask containing highly nutritive medium. And then we are inoculating Streptomyces. And after inoculation, we are incubating it at room temperature. And basically, we are incubating this in a secking conditions, continuous and vigorously secking conditions during this incubation period to allow Streptomyces to produce the antibiotic in an optimum quantity. So here, samples of cultures, it's a liquid culture, are periodically withdrawn aseptically to undertake few routine checkups. So what we perform here? Periodically to check the suitability of different media from maximum antibiotic production, to determine the value of pH at which maximum growth is there, as well as maximum antibiotic production is happening, to check contamination, to determine whether antibiotic produced it's a new one or it's a already established established antibiotic, to check the stability of antibiotic at different pH as well as temperature level, to determine the solubility of antibiotic in different organic solvents, and to check the toxicity of the antibiotic against the experimental animal. Now, after carrying out all this checking or all this routine checking, further we have to study to get some additional information about the antibiotic produced by the new microbial strain. We need to check the effect of incubation temperature and antifoaming agent on fermenter. Basically, now we are trying to check the production level on a large scale. So we will apply the same culture medium, not in an Erlenmeyer flask, conical flask, rather in a small scale fermenter, where we will check the effect of incubation temperature and antifoaming agent during fermentation, rate of resistance developed among the test microorganisms, and we have to check the antibiotic for its bactericidal or bacteriostatic efficiency, its ability to precipitate serum proteins to cause hemolysis of blood, or whether to harm phagocytes. So toxicity checking periodically we have to perform. We have to check for possibility of inclusion of precursor chemical of the antibiotic production in the medium. We have to check suitability of the organism for mutation and other genetic studies. So through this, we can actually develop a general method for the screening of antibiotic producer from soil. So our general outline has been mentioned here. So just we are summing it up, both the primary as well as the secondary screening technique. Here we have collected soil sample. After that, we have diluted it serially, and we have used an aliquot to the agar plate, where in each and every plate the colony forming unit should be 30 to 300. And then we have set the incubation temperature, pH, and time to get colonies over the surface of the agar plate. And then we have subcultured it if we have suspected any zone of inhibition surrounding the colony or colonies. Then we have purified it through stick plate method, and then went for stock culture as well as secondary screening. So the secondary screening to prove that it is non-toxic, it is producing non-toxic product, it is non-pathogenic, it is producing antibiotic with a good yield, biomass, as well as to check genetic stability, to check physical, chemical, and biological properties of the product, to check level of byproduct of the product of the fermentation process. And after all sort of routine analysis, we will go for classification of the microorganism, then comparison of the isolated microorganism with the available microorganism which is available in literature, And afterwards, we can go for strain improvement for better yield as well as better product development. So in this way, we can develop an industrially important microbial strain. So far, whatever we have discussed in this lecture, we have started from the sources. We have discussed about the potential sources. We have discussed about the potential applications of different types of microorganisms isolated from natural sources or man-made sources. And then we have discussed about the isolation and screening technique to establish the isolated microorganism as an industrially important microorganism. That's all.